Option B, Biotechnology, Bioinformatics, Part 1, has its focus on microorganisms and the varied metabolism of microorganisms. This sets the stage for examining the use of microorganisms in industry, the subject of Part 2. The essential idea is microorganisms can be used and modified to perform industrial processes. Here is the outline of all the movies available for Option B. Use this outline to find the movie you need for review. This movie is focused here. Here are the first 3 IB syllabus statements of the movie. Microorganisms are used in industry because they are small and have a fast growth rate. Microorganisms are metabolically diverse. List 4 metabolic processes carried out by named microorganisms. Here is a curve that displays microorganism growth over time. Microorganisms are small and have a fast growth rate. The population grows exponentially early in time, then reaches a plateau where the food source becomes limiting or wastes become limiting. You can see the dead cells increasing through this time period. A couple of things to emphasize. The relatively short time over which the population grows rapidly, and the y-axis is log scale, such that a single unit change on the scale represents a tenfold change in the size of the population of microorganisms. Remember that microorganisms are used in industry because they are small and have a fast growth rate. The metabolism of microorganisms is varied. Autotrophs can be divided into 2 groups. Photoautotrophs use light for energy and carbon dioxide as their source of carbon to produce carbohydrates. Chemoautotrophs use inorganic molecules in redox reactions to generate the energy necessary to transform carbon dioxide into carbohydrates. Chemoautotrophs fall into several groups: methanogens, halophiles, thermophiles, sulfur oxidizers, nitrifiers, and denitrifiers. Chemoautotrophs can have extremely fast growth rates. Some species have the capacity to double the population in an hour. Now let's go to heterotrophs. There are 2 types of heterotrophs. Chemoheterotrophs derive their energy and carbon from organic molecules. There are microorganisms that use light as an energy source and organic molecules for their carbon because they cannot use carbon dioxide as their source of carbon. Spend some time studying the terms on this slide. So let's look at 5 metabolic processes carried out by microorganisms, starting with photosynthesis. Photosynthesis transforms carbon dioxide and water into carbohydrates using solar energy. You know many examples of macro photosynthetic organisms. Work to learn the name of 1 or 2 micro photosynthetic organisms, such as Anabaena, Chlorophyta, or Spirogyra. Nitrogen fixation is the transformation of gaseous molecular nitrogen, N2, into ammonia. This should be review for HL students from your study of the nitrogen cycle. 2 organisms that perform nitrogen fixation are Rhizobium bacteria and many species of cyanobacteria, otherwise known as blue-green algae. The bacterium Rhizobium associates with the roots of legume plants in a mutualistic relationship. You can see the nodules with Rhizobium here. Nitrogen fixation, conducted by Rhizobium, releases ammonia and ammonium to the pea plants and to the soil, while the pea plants provide photosynthetically derived sugars to the Rhizobium. Anabaena is a microorganism that photosynthesizes and fixes nitrogen. These cells are photosynthetic, while these larger cells, known as heterocysts, conduct nitrogen fixation. Fermentation is the process in which a substance breaks down into a simpler substance, and we tend to associate fermentation with the production of alcohol. The term fermentation will be used more broadly in this unit to describe various metabolic reactions in which large organic molecules are broken down into simpler molecules. Members of the fungal kingdom are the best known fermenters. Saccharomyces cerevisiae, seen here, is yeast for making beer, wine, and bread. Aspergillus niger for making citric acid, and Penicillium notatum for making penicillin. Here is the Aspergillus niger used in producing citric acid for food flavoring and the preservation of foods. Penicillium notatum, seen here on this orange, produces the antibiotic known as penicillin. Penicillium camemberti ferments milk to camembert cheese, and Penicillium roqueforti ferments milk to blue cheese. As I mentioned, we will define the term fermentation more broadly than just the metabolic reaction that results in alcohol. Fermentation is the process in which a substance breaks down into a simpler substance, an enzyme-controlled chemical breakdown of an organic molecule, anaerobic mostly, but not always. The chemical breakdown of a substance by bacteria, yeasts, or other microorganisms, typically producing carbon dioxide and heat, but not always carbon dioxide. The fermentation process used in making alcohol starts with glucose. Glucose is transformed to pyruvate in glycolysis. This should be review from your study of respiration. Under anaerobic conditions, pyruvate is transformed to alcohol, ethanol, with the release of carbon dioxide. The fourth metabolic process of the movie is methanogenesis. Methanogenesis is the production of methane gas from carbon dioxide under anaerobic conditions by microorganisms, typically from the archaea domain. Methanosarcina and Methanococcus are examples of microorganisms that perform methanogenesis. The anaerobic production of methane starts with carbon dioxide, runs through a complicated pathway involving various intermediate molecules and ending with methane. Methane is often called biogas. The fifth metabolic process performed by microorganisms introduced in this movie is denitrification. Denitrification is the transformation of inorganic nitrogen compounds such as nitrate to molecular nitrogen gas, N2. The reaction provides an energy source to denitrifiers such as Pseudomonas denitrificans. All of life is divided into three domains: bacteria, archaea, and eukarya. The archaeans are an interesting group as they include the methanogens, the halophiles, and the thermophiles. Methanogens live in anaerobic environments. Halophiles exist in regions of high salt concentration, and thermophiles tolerate high temperatures. The metabolism of archaeans is as diverse as the habitats they occupy. Archaeans are sometimes known as extremophiles because of the extreme conditions that they can tolerate in their habitats. In this photograph, we can see species of salt-tolerant archaeans providing color to these ponds. Marinobacter is a halophile capable of degrading benzene. Benzene is a carcinogenic molecule found along with spilled oil during the extraction of oil from the ocean. Oil harvesting in the ocean results in salty water contaminated with hydrocarbons such as benzene. Humans now use Marinobacter to decompose hydrocarbons such as benzene, and this process is known as bioremediation. Pseudomonas putida is a rod-shaped soil bacterium with a very diverse metabolism, including the ability to degrade oil or methylmercury in the process of bioremediation. Methylmercury is particularly dangerous if it enters the food chain. Pseudomonas putida has been genetically modified with a sequence of genes known as the MER operon. The genetically modified Pseudomonas putida, located in columns, is able to digest the methylmercury. Once it's poured through the column, the mercury can then be extracted and disposed of properly once the solution is moved through charcoal. Pseudomonas putida is the first patented organism in the world. Because it's a living organism, the patent was disputed and brought before the United States Supreme Court in the historic court case Diamond versus Chakrabarty, and the inventor, Ananda Mohan Chakrabarty, won the case. Pseudomonas putida is a gram-negative, rod-shaped soil bacterium used in bioremediation. Bioremediation is a waste management technique that involves the use of organisms to remove or neutralize pollutants from a contaminated site. Pseudomonas putida is sprayed on oil spills in order to digest the hydrocarbons found in oil. We have two small concepts left as we head toward the end of this movie, and the first concept is defined by this IB syllabus statement. Describe the process of gram-staining bacteria, including both gram-positive bacteria and gram-negative bacteria. Based on the structural differences in the cell wall and membranes of certain bacteria, we can group bacteria into gram-positive and gram-negative types. The gram stain is almost always the first step in the preliminary identification of a bacterial organism. Let's take a look at the structural difference between gram-positive and gram-negative bacteria. Gram staining is a method used to differentiate bacterial species into two large groups, gram-positive and gram-negative. Gram staining differentiates bacteria by the chemical and physical properties of their cell walls by detecting peptidoglycan, which is present in high quantities in the cell wall of gram-positive bacteria. The cells are exposed to crystal violet stain, then washed with an iodine solution, then washed with safranin. Gram-positive cells stain dark with the crystal violet, while gram-negative cells stain light pink due to the safranin. The bacteria must be heat fixed to the slide as the first step. Then rinse the cells with crystal violet. This is the primary stain. Then rinse with iodine solution. This fixes the crystal violet to the cell walls of the cells that have been fixed to the slide. And then rinse with alcohol to decolorize. Any cells that do not have a wall will have the primary stain removed. And then rinse with some water. Rinse with safranin, which gram-negative cells take on to provide a pink color. This allows them to be distinguished from the dark purple coloration of the gram-positive cells. Once again, heat fix the bacteria to the slide, rinse with crystal violet, then apply iodine to fix the crystal violet to the cell walls of the gram-positive cells, then wash with alcohol to remove the stain from cells without the wall. You can see that here. Then wash with safranin to color the gram-negative cells pink. Gram-negative bacteria have a double membrane with the peptidoglycan layer, a thin layer, sandwiched between the two membranes. Gram-positive bacteria have a thick peptidoglycan layer outside the cell membrane. Gram-positive bacteria retain the crystal violet dye and thus are stained dark violet. Gram-negative bacteria do not stain a dark color and retain a pink color due to the safranin. Again, gram-positive bacteria have a thick layer of peptidoglycan, the cell wall, outside the cell membrane. The peptidoglycan retains the crystal violet stain, becoming dark purple. You can see the dark purple gram-positive bacteria in this micrograph. Gram-negative bacteria have their peptidoglycan wall sandwiched between two phospholipid bilayers. Gram-negative cells do not hold the crystal violet stain and instead display as pink, seen in this micrograph. Once again, here's the structural difference between gram-positive and gram-negative bacteria. Notice the thick layer of peptidoglycan outside the membrane of gram-positive bacteria. The peptidoglycan stains purple. You should take some time to study this slide. In this micrograph, you can see gram-positive bacteria here and gram-negative bacteria here. And the last microbiological concept of this movie is defined by this IB syllabus statement. Analyze experiments showing zones of inhibition of bacterial growth by bactericides in sterile bacterial cultures. In the 1920s, Alexander Fleming accidentally discovered that the mold Penicillium produced an antibiotic substance that Fleming called penicillin. Penicillin is a bactericide. It kills bacteria by inhibiting bacterial cell wall formation. Howard Florey organized a team involved in the mass production of penicillin, and that team included Ernst Chain. Fleming, Florey, and Chain received the Nobel Prize in 1945. Fleming had an agar plate with bacteria which had developed a growth of the mold Penicillium. The story goes that he was throwing the plate away because of the mold, but he noticed a zone near the Penicillium mold that had no bacterial growth. This led him to investigate the properties of the mold Penicillium, and a metabolic product of the mold was discovered to have antibiotic bactericidal properties. We know this product to be penicillin. Today, we assess the effectiveness of an antibiotic, such as penicillin, by soaking a 6 mm filter paper disc with an antibiotic solution of known concentration. The disc is then placed onto a plate that has been inoculated with bacteria. The plate is incubated in an oven at 35 degrees for 24 hours. After 24 hours, the zone displaying no bacterial growth is measured. The zone of no growth is known as the zone of inhibition. The diameter of the ring, once measured, is compared to known standards for antibiotics. The 6 mm filter paper disc is soaked in an antibiotic solution of known concentration and placed on an agar plate with an inoculation of bacteria. You can see the bacteria here and here. After 24 hours, the diameter of the ring, the zone of inhibition, is measured and compared to standards. The size of the zone of inhibition depends on a number of factors, including the rate of diffusion of the given drug in the medium, the degree of susceptibility of the organism to the drug, the number of organisms inoculated on the plate, and their rate of growth. It is essential, therefore, that the test be performed in a fully standardized manner. Here is an agar plate with bacteria where 8 6 mm discs have been placed. The discs have been soaked with different concentrations of antibiotic solutions. This disc was the control, soaked only with distilled water. You can see that the zone of inhibition increases with each disc in this order. The concentration of antibiotic was increased with each disc from here to here. This graph displays the effect of different concentrations of ampicillin on the mean zone of inhibition. As the concentration of ampicillin increases, the mean zone of inhibition increases. The zone of inhibition at 0.6 micrograms of ampicillin per milliliter is significantly larger than 0.2 micrograms of ampicillin per milliliter. The zone of inhibition at 1.2 micrograms of ampicillin per milliliter is significantly larger than 0.6 micrograms of ampicillin per milliliter. We can test whether bacteria are resistant to antibiotics using the technique of assessing the zone of inhibition. For example, if there is no inhibition, the growth of bacteria would extend right up to the rim of the disc on all sides, and the organism would be reported as resistant to the antimicrobial agent in that disc. This would be the case in antibiotic A, C, F, and D. Now you may wonder about disc D, which displays a zone of inhibition. A zone of inhibition, like around disc D, does not automatically lead to the conclusion that the bacteria was susceptible to the antibiotic being tested. The diameter of the zone must first be measured in millimeters and compared for size with values listed in a standard chart. Here's a standard chart against which measured zones of inhibition would be compared. For example, if you're testing penicillin At this dosage on Staphylococcus, the zone of inhibition can be up to 28 millimeters wide, and the Staphylococcus would be considered resistant. If the zone of inhibition is 29 millimeters or larger, the Staphylococcus would be considered susceptible to the penicillin. Testing resistance to erythromycin at a standard dosage, the ring of inhibition can be up to 13 millimeters, and the Staphylococcus would be considered resistant. In this last slide, we can see the impact of mercury ions on bacterial growth. Heavy metals like mercury pose a real danger to organisms. Mercury ions have been placed on the filter paper discs. Let me direct your attention to the middle plate first. The zone of inhibition in the middle plate is large and representative of the impact of mercury ions on bacteria. The mercury ions have created a large, clear zone of inhibition. The plate on the left displays the response of genetically modified bacteria. A sequence of genes known as the MER operon has been inserted into the bacteria using a plasmid. These bacteria are resistant to the mercury, as can be seen by the small zone of inhibition. Interestingly, the bacteria with the MER operon can digest ions of mercury, reducing their presence in the environment. Bacteria with the MER operon serve to detoxify mercury compounds. The use of bacteria to detoxify the environment is known as bioremediation. The plate on the right has bacteria with the MER operon, but a mutation has knocked out one of the genes in the operon. The resistance to mercury no longer exists, and the zone of inhibition is much like that of the control. And that brings us to the end of Option B, Biotechnology, Bioinformatics, Part 1. Part 1 has focused on microorganisms and metabolism. Part 2 will focus on microorganisms in industrial fermentation.